Hi, everybody, welcome back from coffee break. It's now my distinct pleasure to introduce Georg Wallman, or as he asked me to call him for 10 months, George. And as I called him, Jorge, whichever you like. So Georg has a long history with SCP and the Slavov lab. So he came to us for his master's thesis and was with us for about 10 months. And it was a very, very, very successful, very good time, I think, very productive. Georg authored a paper sort of extending the DoMS framework for DIA data. While doing that, he also got into understanding how search engines work, feature identification, and many more things. After his thesis was done, of course, Georg moved forward and went to the Mon Lab at the Max Planck Institute, where he's been a PhD student now for a couple of years. And he has, again, continued a lot of computational workflows. His sort of main publication was alphaDIA, which is an open source, I'm calling it main, hopefully you agree, alphaDIA, which is an open source search engine end-to-end that uses transfer learning. And yeah, today Georg's going to tell us about exploring the limits of large-scale DIA search. Looking forward to your talk, Georg.
Thank you so much, Sad, for the introduction. Yes, so I'm a PhD student at the TS Mans lab, working on all sorts of problems involving DIA search and how we can improve it, how we can make it accessible to the community. And part of this is also kind of adapting to the changes we've seen in the field of proteomics over the course of the last years. And this is that we have achieved substantially higher throughput, so throughput to a level which we wouldn't have imagined a couple of years ago. So more than 100 samples per day measured on a single instrument. We have these very sensitive TOF detectors like the Astral and the timsTOF, which allow the very short gradients, very deep proteome coverage. And this has resulted in quite large cohorts and larger, ever larger data sets. So be it single cell proteomics, but also plasma studies, tissue studies, where we start seeing more than 10,000 samples being quantified in the same run. And single cell proteomics and also kind of the projects and data sets we've seen today is a major driver. And this brings the challenge about how we can continue processing these data sets and how we can, adapt the tools we need to not hit a roadblock at some point. And I can imagine most of you had a large study, maybe a couple hundred samples, maybe a couple thousand samples. You put them on some Windows computer, some workstation. You started to search, and after 4 days, 5 days, 6 days of work, it crashed out of memory. And you then just went forward, did it again. Maybe you were lucky, maybe you bought a bigger computer. But of course, this is a work mode which doesn't scale. And so what I'll be talking about today, kind of very briefly for the last session, is to give you some perspective how DIA search works, kind of what steps happen in between, and how all of this translates with computational complexity and how we can adapt it for distributed search so that we are not limited anymore with the number of samples we can process. And in the end, I will give you the perspective how we've used this with some of our largest cohorts, which is like a plasma study, which is about to be published with the Bill and Melinda Gates Foundation, where we've processed and quantified more than 40,000 proteomic samples, so multiple years of Astral measurement time in a single run, successfully with label-free quantification. So the platform we are using is alphaDIA, as Sad introduced. So it's an open source DIA search engine, which is user accessible, which allows kind of everybody to process their files. And we've released the first version as a preprint last year. The paper will come out briefly or shortly. And we are kind of continuously developing like an open ecosystem, which where we want to, of course, offer cutting edge tools, but where we also want to make transparent how the processing works. And we want to offer kind of the community the tools that they can adapt and improve existing methods. And that kind of, especially with DIA, where this data is so complicated, we move away from this black box processing approach. And what alphaDIA gives us is full control over the processing workflow. So we start with the raw files at the beginning, be it from the SciEx instruments, the Astral instruments, or also from the timsTOF instruments. We have the spectral library prediction with the package PepDeep from our alphaX ecosystem. We handle libraries, add annotations, add modifications, perform mask calculations, and then throw everything into alphaDIA. And following search and FDR correction, we end up with our FDR controlled precursor list, and in the end get our protein quantities from direct LFQ. And one of the nice things is that this runs on all, it's mostly Python based with some kind of high performance improvements, and it runs on all different infrastructures. So we can run it on Windows, on Linux, and we can also run it on the cluster. And the story I'll present you today is how we've adapted it for very highly parallelized distributed cluster use. So how we can, the example I will be using, So what are the steps which we have in DIA search? The first step, it's kind of obvious. We know that most search engines do it by default. If we're talking about peptide-centric search, in a way how Diane performs it, Spectral performs it, or also alphaDIA performs it, we start with in silico library prediction. So we're building a library of 2 or 3 million precursors potentially detectable based on a FASTA file. Happens once per analysis, and it's usually a pretty fast process, takes maybe 20, 30 minutes, and the output is our spectral library. We then go on, and then kind of the computational complexity starts using a first search with a fully predicted library. So this means that we are quantifying and extracting precursors at 1% local FDR, and we have to perform this process once per raw file. And this is usually what is the computational bottleneck if we perform large-scale analysis, because it's performed sequentially. So if we have thousands of files, we have to wait for one file to finish for the next one to begin. So we went with a modularization approach where we could do these searches all in parallel, and the precursor quant, which is then generated in the end, could be collected. This precursor quant is then collected in the third step, which is a consensus step, which means that we start gathering all of this information, all of these quantified precursors, and combine it in an MBR library. And at this step, we also perform the protein inference, we perform false discovery correction, and we perform spectral library building for the next step. What is the challenge about this? Computationally, gathering all of this information is not very challenging, but we have to load everything at the same time into memory. So we are limited by space, not by time. Then we go into the second step of the search, which is the MBR search, with the goal to reduce the missingness in the data and to increase the completeness, and also to help identification of locally abundant peptides, especially in the context of single cell, or in the context of single cell or plasma. We perform this step again once per raw file, so we have the challenge that we have to distribute this load, but because this is somewhat independent, we can take the same strategy as from the first search, take different quant files, and then kind of combine them in the final consensus step. And here the challenge is really that we want to perform the label-free quantification, where we compare kind of like with like. So we compare the same precursor across all different samples, and this means that one last time we have to load everything into memory at the same time, and we are limited with space complexity. We went forward and implemented this distributed search approach, and we have the tools online. You can check them out if you want to get started yourself to parallelize this in a cluster or in a cloud setting yourself. So you can use it with your Slurm cluster, your research cluster, to have a search which runs much faster and can also be continued if it stops in between. So we went forward to test this with the project of a colleague of mine, Marvin Thielat, who's also here today, who's working on some of our single cell data sets, using a data set of 1,670 single HeLa cells, and acquired or processed with the Cellin1, then measured with the Wisp-Zoom gradient ATSPD on the Orbitrap Astral. And this data set, couple of hundreds of gigabytes of data, we wanted to process on our local cluster with 50 different nodes, and then 2 jobs per node. And this wasn't any kind of high performance computing setup. You can imagine if you have such a cluster and you clog it up with jobs for the next 2 weeks, your colleagues will be mad. So we actually used kind of the oldest partition of that cluster. So it's only 10 cores, 10 different CPU cores per job, and 64 gigabytes of memory. So it's not that you need like the most up-to-date hardware to do this well. So this is what we used. So for the first step, predicting a spectral library, as I mentioned, it's kind of sequentially. This was easy to do. So this would have taken, or this took 20 minutes in the whole workflow. But then kind of the major bottleneck was, as I mentioned, the first search. Within the first search, what we are doing is we take the library in a peptide-centric manner. We look for each and every precursor if there's evidence in the data. If we can score the precursor, if we can gather the evidence for that precursor, and if it then kind of survives in the FDR competition. And what we see is that this step consists of two different sub-steps. The first one is that we have to identify all of the hyperparameters. This is what I name, or what I call here, a search space optimization, where we calibrate the retention time predictions, where we reduce the retention time limits, where we reduce the mass limits you need to quantify a precursor with a given mass accuracy. And what we see is that these steps on average took 20 minutes per single cell. And if we look at the correlation between the duration of the optimization step and also the number of proteins we got, we see, interestingly, a clearly negative correlation. So if you have lots of input material in a single cell and you can identify a lot of precursors, it's much faster to perform the step. But the lower we go, the fewer proteins we find, the longer it takes for the optimization. And this is something you might know kind of intuitively from running searches yourself, that the fewer material is in your sample, the longer it takes kind of for example, Diane to identify anything, and you can end up kind of with it just searching for hours in the same data. In the end, kind of the quantification was pretty fast, was like 8 minutes per cell for the whole library. So we ended up with a relatively slow process of 28 minutes per cell. But because we had used 100 different jobs in parallel, we were able to use to search 3.7 cells per minute. So we were able to search the whole data set in 7.5 hours. Then kind of we started the first gather operation, where we created the match and run library of all precursors, identified the 1% FDR, and we went into the second search. And as the search space was already reduced very much at this stage, so we were only looking for 40 to 50,000 precursors across the whole data set, it was much faster and much easier to quantify. So the search space optimization, where we found the right search parameters, only took around 2 minutes. And you can see that on average, we quantified around 4,000 proteins in these single cells. And the quantification itself, the extraction of the quantitative information, only took around half a minute. And this also tells us that the second step in this two-step searches is so much faster, which means even if we have 1,000, 2,000, 5,000 cells, it might make sense to perform the library building with a spectrum-centric tool like MSFragger, or only with a subset of the library, which still kind of exhaustively covers all precursors which can be potentially identified, and tend to use only this fast peptide-centric search for the quantification. So kind of processing this whole data set with that library only took 60 minutes in total. And if we would now reprocess it, for example, with an existing library or in a library which was empirically acquired, this is something you could easily do kind of with a click of a button, and you would have the results 1 hour later. And kind of speeding this process up, of course, gives the researcher then the ability to iterate much faster and test hypotheses and find out what issues there might be with the data processing. The last step was then the label-free quantification, which kind of was pretty straightforward, took 50 minutes. So we were able kind of to process this large data set in around 10 hours of total time. So how far does this scale? As I brought up, all of these different processes can be slow, but we can parallelize them pretty well. But if we do this, there's another issue. And this is that these steps in between, so these kind of consensus steps where we combine all data, can be a limiting factor. And the reason for this is that we have to combine all of the information on one machine, which means that we might be limited by memory. And when we worked on this large plasma proteomics study with more than 40,000 samples, we actually weren't able to quantify everything in Diane, even if we used a computer with like a terabyte of memory, because it was just beyond to kind of what you could gather on a single machine. So we looked into our internal LFQ implementation, direct LFQ, of a colleague of mine, Konstantin, and we realized that that how LFQ works can actually be exploited to scale further, even to these very large studies. And the idea is that during search, we parallelize across these rows. So if you think about the matrix you want to get out of the quantification, the LFQ matrix, we have a matrix of individual runs as rows, and then individual proteins as columns. And what we're interested in as an output is the protein quantities in that matrix, and as an input, the different precursors and fragments which give quantitative information to this. So during search, we can easily just look at one slice across the row, and we don't have a very complicated problem because we only look at one file at a time. But then kind of during classical LFQ, we load everything into memory. At the same time, we only, for quantifying a single protein, we only need the information from that single protein across all different runs. So we went with a vertical slice and adapted our internal direct LFQ workflow to operate on single proteins at a time, or batches of proteins at a time, a couple tens, a couple hundreds, and then kind of combine this information in the end into a single table with a delayed normalization kind of to account for different total intensities or different kind of ionization efficiencies. So this turned out to work very well, and also kind of to be not to scale much beyond what might be 40,000 samples. So this is something which will be then published, or which is about to be published with the manuscript for that plasma study. So what is the takeaway? We can scale these proteomic workloads optimally, facilitating ever larger cohorts, even going to tens of thousands of samples. And the takeaway is also that library building optimization are still computational bottlenecks in such a DIA search workflow. So the search itself is somewhat simple, but finding the right parameters, understanding the data your instrument has produced and how it was produced, can be challenging, and also the label-free quantification, has been a memory bottleneck, but we think we have found a solution how to scale it out of memory, kind of. So yes, all of this has been a collaboration with friends and colleagues from our lab, working on kind of bioinformatics topics, but also on some applying these methods, and we're also supported by lots of sponsors. So I'm open to any questions you might have.
So I have a technical question about your second pass search, which you call match between runs. Typically, match between runs means that you only use MS1 information. Here, it kind of sounds like that's not the case.
Yeah, it's a very good point. So I use the term match between runs in the same way as it's used by Diane, for example, in a way that you have a focused library.
It's a two-pass.
It's a two-pass search. Yeah, okay. I feel like this is one of the kind of the naming issues which have persisted in the field, and then somebody started calling it match between run, and it has kind of sticked around. It's a very good point.
Hey, Georg, as always, very clear presentation. When it comes to the building of the spectral library, the optimized spectral library for very large data sets, let's say that we analyze 10 million single cells, it seems to me that at some point there will be a sub-ensemble of the cells that is large enough to give you all the benefits of building that spectral library, so that we don't need to build it from every single cell that we analyzed. We can just take a group of maybe 2,000 or 5,000 single cells, and that will give us all the benefit if we sample it. So maybe that's one approach of determining at what point the marginal gains of including more runs in optimization are beginning to level off, and it's not worthwhile pushing the limits on memory.
That's a very good point. So this is for sure one strategy, and the question is, when do you start leveling off? And one thing we've seen, I don't have the plot on the slides, but we've done this experiment with this very large cohort of 40,000 plasma samples. And interestingly, we see that it never really levels off and becomes flat, but it kind of keeps increasing at a very slow pace. So we think that there are some FDR issues hidden, at least with the tools we've used, which in this case was Diane, on the protein group level, which only emerge, or which only manifest itself if you go into the range of thousands, maybe tens of thousands of samples. But there's also another answer to kind of what this number can be. And this is, we all use data completeness filters before we process our data. So for example, we filter our proteins to 60, 70, 80% data completeness. And this also kind of gives a natural, I mean, you can do the statistics. If you have an 80% data completeness filter, you can, after 10 or maybe 20 samples, it becomes highly unlikely that you haven't covered any of these proteins just by random chance. So the number of samples we need for generating a library can be surprisingly low. I think even like a 20% of the samples can be sufficient. Yeah.
Thank you. And I have another quick question. I know that you have thought a lot about, and I think it would be of benefit to this community to discuss. And namely, we optimize the parameters of our spectral libraries on the same data that then we go and research. And there is this possibility of circular logic. And I know we all want to avoid that, and you have thought about it. So if you can share with us your thoughts of what do you do to avoid that?
Yeah, that's a very good point. So what I think I think what's important to use is use the best practices from machine learning. So when we try to apply these methods from machine learning, we also have to use it's not only about kind of using all of the amazing libraries and all of the capabilities, but also kind of taking the how to put it, like the necessary kind of steps you have to take. And this is using, for example, kind of meaningful and good test and train and validation splits to make sure that the data you're validating on, or that you're validating on a portion of the data you haven't used in the training of the model. This is one thing. The other benchmark we've run a lot with the transfer learning, for example, is that we've used transfer learning in combination with entrapments. And we've included the false positive identifications from the entrapment search in the transfer learning training data to see if they are being memorized in the next step for the library prediction. And even if we do multiple iterations of that transfer learning, we see that the number of false positive identifications in the entrapment FDR actually goes down and not up. And most likely for the reason that for these false positive identifications, the model cannot generalize, and it needs the capacity of the neurons to model the real true behavior of the peptides, where it can lower the loss compared to overfitting on some spurious data. Yeah.
Thank you.
So for building the spectral library, because we are starting with a theoretical spectral library, then in the first pass, we are making a library from the data. So my question is, what is the need of keeping a quantitation at that point? Because in generally for other softwares, when you build a spectral library, you never have a quantitation values there. So what is the need of there? And if you do a quantitation, like is it like MS1 level quantitation or MS2 level quantitation, or keeping a both? And I mean, what is the need of, because anyway, we are quantifying the peptides at the second level. So can we reduce the time?
Yeah.
Because
It's a very good point. So we don't need any protein or peptide level quantification. We need precursor level quantification, for example, for predicting the charge state and fragment level quantification to train models for the spectral library. But we don't need any protein level quantification. That's so. This was maybe kind of a bit not quite clear, but kind of the LFQ is only needed in the last step and not in the middle. Yeah. Thank you.